Hello, and welcome to GSEA Background. My name is Laura Harris, and today we will be exploring gene set enrichment analysis. So let's get started. To better understand GSEA, we need to understand a little bit about the type of data that GSEA calculates on. To understand that, we need to define some biological terms. We'll start with a gene, which is a piece of DNA that is the basic physical and functional unit of heredity. We always talk in genetics about passing your genes on to the next generation. Genes are stored in DNA. DNA is a permanent molecule. DNA is what gets transferred from parent to offspring. DNA contains many genes. If you're a bacteria, this could be a few thousand, could be 30,000 if you're a human, or more if you're plants. Whenever a cell wants to express or produce a protein product from a particular gene's instructions in the DNA, it makes a copy of that particular gene in the DNA in terms of a messenger RNA or an mRNA. This copy then is what's used to do translation in order to make a protein product. This protein product is then used in all sorts of cellular structures and processes for cell survival. So when we look at gene expression data through GSEA, we're really looking at the simultaneous profile of the expression of 50,000 or so genes, depending on what my model is. So you'll see, for example, gene expression data in the lower right corner, but how do we get that data? Well, we actually get those numbers from taking messenger RNA samples from different conditions. In the example for this class, we'll be looking at SARS infected versus mock infected. So we'll take RNA from SARS infected samples and RNA from mock infected samples. We will tag some complementary DNA that is made from the messenger RNA we collect from our cell samples. And then we undergo some sort of an analysis. In this case, we're looking at microarray, which is a hybridization to a chip. But regardless, we're looking at how things fluoresce or how things bond to a known molecule, and then we can use computational analysis to quantify what we see in terms of color. This is the data you acquire from something like Gene Expression Omnibus. So what do we do with the data we get? Well, first off, we have to identify where that data came from. So some samples are going to be SARS infected, some are going to be mock infected. For our analysis here, we're going to use differential gene expression. Differential gene expression is when you compare between your SARS infected and your mock infected, or whatever your experimental conditions happen to be. In this particular case, we can do that for each gene individually that we measured by using some sort of a statistical analysis like a t-score. So then you'll end up with what's called a gene signature, which is a ranked list of genes based on the differential expression as defined by an appropriate statistic, such as a t-score, as you see here, fold change, etc. You can visualize this, and GSEA does this for you as part of its enrichment plot, where we see red indicating overexpression of a particular gene in SARS infected versus mock infected. Blue at the other end is underexpression, so these are genes that are higher in expression for mock compared to SARS infected. And in the center of our gene signatures usually are genes that are equal between SARS infected and mock infected and have a t-score around 0. What is single gene analysis? Well, as we just saw on the prior slide, we can look at genes individually as part of a gene signature. So we can do that by ranking by a particular statistical method like we saw t-score, or if we want to be more selective, perhaps we'll use two different statistical analysis. So here on the right, you see an example of a volcano plot. You have the log fold change, on the x-axis, and you have the negative log of the p-value from your t-score test on the y. If we establish some particular cutoffs, so for example, a t-score p-value of less than 0.05, or perhaps a fold change that's going to be greater than 2. Yeah, I'm seeing error here, so I'll go ahead and fix that in your handout. But anyway, as you see on the slide, we can then designate certain groups to be genes of interest. So here I've got the blue and I've got the red dots to represent the under and the overexpressed, respectively. So what is enrichment analysis? Well, now that we've looked at which single genes meet specific criteria for us to be interested in them, we can look at things like what do they do. So in order to get an idea of what these particular genes of interest, say for the red group only, might be involved in, we can do an enrichment analysis where we compare the membership of the genes in red to the membership of a known gene set, say for a particular pathway like glycolysis. And then we can see if the proportion of genes between the two data sets is going to be greater than what we would expect by random chance. So by doing that, we can use two types of enrichment analysis. There's the Fisher's exact test, which is the most commonly known enrichment analysis, but there's also gene set enrichment analysis, or GSEA. GSEA has some distinct advantages over Fisher's exact test, which is why it's used in your project for this course, so we'll talk about that right now. So what is Fisher's exact test? Just in case you haven't heard of it or you're a little foggy on its details. It's been a while since stats class for some biologists, I feel ya. So the objective of Fisher's exact test is to assess the significance of overlap between signature and query set. So here's a visualization. Let's say I have a gene signature that has 2,000 genes. We'll represent that as the gray box. 200 of those genes meet my particular cutoff. Say my p-value is going to be less than 0.05. That can be the blue circle here. Then I have a query set. For example, genes associated with glycolysis. There are 50 genes associated with glycolysis in this theoretical example. So you see that as the green circle. And 10 genes overlap between the 200 items that I found in my cutoff and the 50 items in the query set. So you see the 10 overlap. And the trick here with Fisher's exact test is that it only considers the genes in the signature that meet a particular cutoff. So it will only think about the 200 genes in that blue circle. It ignores the 1,760 other genes hanging in the gray box. And this is a challenge because some of the genes in that gray box might have been close to meeting that cutoff and become part of the blue circle, but they still have some sort of a biological relevance, and we don't want to miss them because of statistics. So then what is gene set enrichment analysis? Well, gene set enrichment analysis also looks at enrichment. So how over or underrepresented a particular group of genes is in a particular data set. In this case, though, GSEA considers the whole signature, even the genes that do not meet a particular cutoff. So since GSEA will consider the whole signature, you get a more global perspective. So let's see the details. So here in the center I have an enrichment plot. You will see the reference signature, which has all of the genes in that particular set. In this case, I've got an actual data, so it's around 5,000 or so genes. These are ranked by differential expression. Remember, red is high for SARS infected, and blue would be for mock infected. What you see here is hit location. So this is where on the gene signature we get matches with the query set. And you'll notice that a lot of the matches are clustered at the red end, or what we call the positive tail. Now, GSEA will calculate an enrichment score, this green line here. The enrichment score goes up whenever we have a match, and it will go up by a score related to whatever its rank is. So whatever the t-score or the fold change value is. GSEA will have an enrichment score go down every time there's a miss, and that is based on a constant that's related to the size of the query set and the size of the reference signature. That way you always start at 0, and at the end you end at 0. And you'll notice we keep going up because we keep getting hits in the positive end. Then when we start to hit misses, we start to go down. So there's a maximum enrichment score for every GSEA. Once you hit the maximum enrichment score, you then start to go on down, and all of the genes that are contributing to reaching that maximum enrichment score are considered leading edge genes. So these are the ones that are most likely going to be involved in whatever biological process, in this case SARS infection, we are looking at. Now we can look at statistical significance in terms of a random model. So once GSEA has done this for your actual query set and your actual reference signature, it then will randomize the genes in the reference signature, but keeps your ranking metric, in other words, your t-score or your fold change value, the same. It then will rerun the analysis, come up with a new maximum enrichment score, and do this 1,000 times in order to establish a null distribution. From that null distribution, the gene set enrichment analysis program will calculate a normalized enrichment score and a nominal p-value. These are important because this will tell you how likely you are to get your actual enrichment score by random chance, and these NES and p-values are what's reported in the literature. So here are some examples. If I have positive enrichment, I will see my items or genes clustered towards the red end of my signature. I will get a positive normalized enrichment score and a p-value less than 0.05. If I have negative enrichment, I will see my genes clustered in the blue end of my signature. I will receive a negative normalized enrichment score, but my p-value will also be less than 0.05. If I have no enrichment whatsoever, I will see the genes somewhat evenly dispersed throughout the signature. My normalized enrichment score will be somewhere around 0, and my nominal p-value will be greater than 0.05. Okay, so this shows significantly enriched query set items, genes, will cluster at one end or the other end. So just to summarize the advantages of using GSEA over Fisher's exact test. GSEA considers all the genes in a signature, not just those that reach a cutoff. That makes it more sensitive to identifying query sets where several items may not reach statistical significance, but collectively have some sort of a biological relevance. GSEA is used extensively in human models like cancer, but is very rarely used in microbiology for several reasons we won't get into here. So just to show you the power of GSEA over Fisher's exact test as a visual, we'll go back to our original example, except in this case my signature has around 5,100 genes. 600 of them meet my particular cutoff. And I've got a query set that's got 200 genes. 32 of those genes overlap with my 600 genes that meet the cutoff. If I run Fisher's exact test on this particular condition, I actually will not achieve statistical significance. My p-value is greater than 0.05. And as a result, I will say that there is no enrichment. I will then reject my null hypothesis. However, if I use the same data for gene set enrichment analysis, you will notice that because Fisher's exact test only takes genes that meet a particular cutoff, it only takes a certain portion of all genes that are considered in the leading edge by gene set enrichment analysis. Because gene set enrichment analysis will consider these borderline genes in the orange circle, where Fisher's exact test only considers the ones in the yellow circle, gene set enrichment analysis will detect enrichment in this data set. And so you'll get a positive normalized enrichment score and a p-value less than 0.05. So this illustrates in a legitimate example how GSEA can be more sensitive than FET. The other really cool thing about GSEA is that it's user-friendly and it's easy to download. You can download it from the Broad Institute at www.gsea/msigdb.org. It's available in Mac, Windows, Linux. You can install it on ICER's HPCC, for example. There's a Java version. You can have R packages, you have Python packages, so it's everywhere, and as I said, relatively used widely with human genomes. So this has been a brief GSEA background. Again, my name is Laura Harris. You can reach me anytime at training@msu.edu, and I look forward to working with you and answering your questions. Have a great day.